Chapter 8, the chemistry in action. So last time we were talking through substituted hydrocarbons. And so as we think of those, I wanted to take a minute and write some things up on the screen for you. So we would call these substituted, and they were hydrocarbons. Specifically, you need to answer in yourself, in your own mind, what is a hydrocarbon? It's a compound that contains what two elements? So you should be saying hydrogen and carbon. A substituted hydrocarbon has a set of things called, or known as, functional groups that are allowed to be substituted in for the carbons. And so I'm going to write a little list here. We'll make this just for your sake of study. These are called functional groups. First of all, as we look at hydrocarbons at all, we need to understand that there are several types, but we know it's going to have a carbon backbone, and it's going to have a set of hydrogens off of it. Now, we said that there was a reason why we studied hydrogen. We studied this in a section called organic chemistry. These are compounds that contain specifically carbon. This molecule is called ethane. And it's called ethane because it has how many carbons? Good. So remember, the prefix is meth, eth, prop, bute, etc. as you go down. And so this is ethane because it has two carbons. As we look at here, there's carbon here, there's a carbon here. The number of H's are not considered when we name the molecule. We get the ane ending because of the fact that there are only single bonds within my atom or molecule. And so we're going to call this ethane. If it had a double bond, we'd have to call this ethene, and we'd have to use a different name. So erasing some of this stuff, what we need to do next is to go through and name each one of these as substituted hydrocarbons. Let's get rid of our double bond as well. We had four functional groups that we talked through. The first one was if we had haloalkanes. Okay, alkanes. Remember, alkanes are single bonds, but haloalkanes have a substituted hydrocarbon that is a halogen. And so simply, all I'm doing is I'm looking at any one of the hydrogens. Doesn't matter which one we have, but any one of those hydrogens can be used. And so looking at this, I'm trying to change my marker on here. Okay. For instance, here we have an H atom. Okay, it's a hydrogen. And so in this case, rather than having an H, I can replace it with any of the halogens. So if you look at the periodic table, you'll notice that you have a set of elements over here. The far right row is called the noble gases. We will ignore that row, but right next to it, group 17, we have a set of things. From the top, you have fluorine, you have chlorine, you have bromine, you have iodine, you have astatine, and so any of these in this group are called halogens. The word halogen means salt formers, and so I can take any one of those, and in this case I choose fluorine and I can put it in there. This is what we call a substituted hydrocarbon, or a haloalkane in this case. I take any of the H's and I replace them with a halogen, I get a haloalkane. But to go back to my original, I would like to show you a couple other things. I could also replace, number 2 here, I could also put in what we called an alcohol. And I specifically underlined 2 letters here, and that was the OH, because an alcohol always adds OH in place of a hydrogen. And so over here I can have an O that is bonded to an H. So in this case we would have, it would be called ethane because it is just a single bond, but we have that ending. If it's an alcohol, we'll add the suffix "all" to the end of it. So we will end up getting what's called ethanol as the name of this molecule. So just reviewing some of this stuff with you so you're comfortable with it. An alcohol, so we replace that portion with it. To go through next, then we had number 3. Let's see if we can get a new color. We had number 3, and number 3 was to have, instead of an alcohol, we had the carboxylic acids. Okay, and that's going to have, if you recall, a COOH. And so where the hydrogen would have been, I'm going to replace that with a carbon, double bonded to an oxygen, single bonded to an OH. And so this right here is what's called the carboxyl group, or the functional group that is the carboxylic head. So we'll talk about that a little bit in just a bit. That's a carboxylic acid. Now the next one's not too far off. I won't change the color because it's very similar. It's called an ester. And an ester has notice the writing here is COO and then another line, because an ester typically is a linkage. What it does is it focuses on the H in the carboxyl group and removes it. When we remove this H from the carboxyl group, we now have a slot where things can go. Typically, this will actually bond in to another molecule. In this case, we could have C's and we could have more H's coming off of them going on. But this portion right here in the middle, this linkage, is what we call an ester. So you have carboxylic acids, alcohols, haloalkanes, and esters. I wanted to remind you of those as far as a chemical shape goes to get you ready. Okay, well, that's what we were looking at this week. But I did want to introduce you again, and we've talked a little bit about it on page 181 of biochemistry. So if you open up your textbooks to page 181, we will look at together biochemistry. So again, looking at chemistry in action, we want to see what chemistry does. We want to talk about why we care, why we'd want to even consider studying this at all. What is biochemistry? Well, organic chemistry is not the same thing. Because I have the pen here, figured I would write a couple things. Biochemistry versus, let's do organic chemistry. Organic chemistry deals with specifically carbon. Biochemistry deals with the root word here, bio, which refers to life or living things. And so this is the chemistry of living things. So in biochemistry, we deal with how things live, how they respond, how living things do chemistry within themselves, whereas organic chemistry deals specifically with compounds containing carbon. So our first point here is biochemistry is the study of the chemistry of living things. And when I think of biochemistry, I honestly go through in my mind a nutrient facts label. I mentioned to you already that biochemistry, honestly, before we even consider biochemistry, we need to think of biology. We know that when we eat food, digestion starts in the oral cavity, in our mouths. The scientific term for that is the oral cavity. As we ingest food, we chew on it, we coat it in saliva, it goes down our esophagus, into our stomachs. It's coated with an acid. The saliva is an enzyme that speeds up the work of that acid. That acid breaks down the foods. It's important that we chew it so that our body does less work. As that food goes into our next portion of our digestive tract, or the alimentary canal, it goes into our small intestine to be coated with more digestive juices. Again, our body is actually absorbing nutrients from that food, but the goal of the digestive tract is to take solid foods, remove the liquid from them as they liquefy, and make them more solid before they leave our body. And so what we know about biology is important, but the question is, what is it that we are pulling out of our food? What is it that we're getting out of our food? Well, if you look at the back of anything, Oreos, donuts, hopefully something a little healthier than that. In this case, I've got nutrition facts of a potato. You will see here that I've got one potato, mentions the calories 110. We have got total fats, you've got 0%, cholesterol, sodium, carbohydrates, proteins, and a bunch of vitamins and minerals. You ought to be familiar with these general terms because that is what biochemistry is. Now, from a biological standpoint, you know these are nutrients, hence the term nutrition. But we want to understand this from a chemistry side. And so today I'm going to spend some time walking through it. The first thing you notice, even back on the slide here, many times is the carbohydrates. This particular one, calories, is large, and most people focus only on the calories. Calories are a part of the energy that we get from food, but also the carbohydrates. So let's talk about that together. Carbohydrates, by definition, are compounds that provide most of the energy for living things. If you had to eat food for energy, I hope you would pick carbohydrates. Foods that are rich in carbohydrates would be something like a potato, something that has a lot of sugars in it, healthy sugars. And so compounds that provide most of the energy for living things. A steak is not a carb, it is a protein, and so we will talk about that as we go. But the word carbohydrates is kind of a special word. When we think of it biologically, we just use the term carbs. We shorten it. But if we look at it from a chemistry perspective, you see the prefix here, carbo, which refers to the element carbon, hydro, which deals with hydrogen in this case. And ates is a suffix that is used for any molecule that has a water atom bonded to the end of it. So this is a substance that is composed entirely of the elements carbon, hydrogen, and even oxygen, carbohydrate. It is typically done in a 1 to 2 to 1 ratio. One of the ones that I mentioned and is one of the most common is this molecule C6H12O6. You'll see that 1 to 2 to 1 ratio. For every 1 carbon you have 2 hydrogens, and so that's what I mean by that. So we'll leave that there for now. So hydrogen, oxygen, carbon are in a carbohydrate. There are examples of this, and when you think of sugars, you often think of bad for you foods, but it is sugar that we are actually eating when we eat foods. Sugars, starches, glycogens, and even cellulose. If I take a moment and go back to the last slide, you'll notice even underneath carbohydrates, carbohydrates have fibers and sugars kind of as a subsection, because carbs are sugars. And so that's what I wanted you to be aware of. So as we go back through this, we know that the examples of carbs are sugar, starches, glycogens, and cellulose. Remember, ose is a suffix in biology that refers to sugars. So as we see ose, as we talk about carbohydrates, think sugars. Well, I have here a glucose molecule, and you'll notice that it is very common for them to form rings. And that's what I wanted you to see, this general cyclic structure as we continue. So what is a sugar? It's a carbohydrate. They're synonymous, but in this case, small groups of carbohydrates containing up to 10 rings of atoms. A sugar is just a ring of atoms, specifically carbohydrate atoms. And so in this case, we have carbon, hydrogen, and oxygen. So we use terms, honestly, it should be, this one slide, I should compare and contrast all 3 of these. The terms are monosaccharides, disaccharides, and polysaccharides. If you take your book to 182, you'll actually see a table there at the bottom. There are some space-filling models of molecules of glucose, sucrose, and even a starch. And you'll see they have rings. The glucose one, which I have a picture here on the screen for you, it's very similar to the one that's in your book of glucose. It's just a ring. A sugar can have up to, notice, 10 rings of atoms. And so what we have here is a monosaccharide. The one that you have on the top left of the picture on 182, bottom of the page, is going to be a monosaccharide. Saccharum is another root word that just simply means sugar. Mono means one. And so it's a one-ring sugar, simple sugar in this case, has one ring. Glucose, we mentioned, is one of those important monosaccharides I want you to be familiar with. One example and then what it is. As we continue, I do want to talk about where that actually comes from. Sugars come from. Well, the most obvious source of this is this process called photosynthesis. Breaking down the word. I know this word is taught for years. We learned this probably all the way back to the 2nd, 3rd grade. But photosynthesis, the first word, prefix photo, means light. Synthesis is to build or to create something. So it is to build or create with light. The question is, what are we making? Well, I hope that at this point you know, I'll write this at the very top, that photosynthesis requires a plant. Specifically, plants are going to take water from their roots plus carbon dioxide that we breathe out using sunlight, to produce 2 things. One of the things we get from plants is oxygen. We have to breathe that. And another product is this C6H12O6 molecule we call glucose, or really simply stated, food. We need this stuff. We need food and oxygen from plants. So photosynthesis is the process in which glucose is made in green plants. We know that each plant is green because it contains a pigment called chlorophyll. As you work your way into it, you'll notice as you cut through the plant, you'll find these stacks inside of a section called a chloroplast, and that's where photosynthesis occurs. It's a process in which a plant takes water, H2O, carbon dioxide, which is in the atmosphere, the heat from the sun, that energy, the activation energy there, to produce oxygen, air, and sugars. So photosynthesis is a process I want you to note. Okay, so you'll notice here I wrote a balanced equation of that reaction here. I mentioned here a disaccharide as well. When 2 molecules of a monosaccharide such as glucose are linked together to form a larger molecule, we get a disaccharide. Again, saccharum means sugar, and di means two. If you look at still on page 182, you'll notice that there is a set of 2 rings for the sucrose molecule, and that is a disaccharide. So if you need a monosaccharide example, you'll look at glucose. If you want a disaccharide example, you'll look at sucrose. All right, continuing on. Sucrose is one of our examples here. A couple of pictures. I just wanted to show you this one. Notice here is sucrose has the molecular formula C12H22O11. In this case, if you look over to the left side, you've got a ring. On the right side, you have a ring. Together, this forms a disaccharide. By itself, it would form a monosaccharide. Another term you might hear in the discussion of carbohydrates is the term polysaccharides. Again, word study. Poly simply means many. Saccharum still means sugar, so it's a many-chained sugar. Notice my definition. Multiple monosaccharides, forgive my typo here, monosaccharides linked together to form polymers. Remember, a polymer is a set of monomers pushed together, sets of individual particles. And these are simple sugars. Well, this really seems like a more efficient use of storage of our food. If we eat only one chain at a time, and our body digests one chain at a time, polysaccharide isn't something we want to digest quickly. It's something we want to store, and we want to pull from those chains over time. Well, there are 2 examples of that I want you to be aware of. In plants take that glucose and they store it in their roots in a form called starch, and we as humans, we store it as glycogen. You can see that in a plant. As a plant grows, you'll notice it has primary growth. The plant gets taller. It's using energy from the food, the starches, to get taller. It also gets thicker. You'll notice that it has secondary growth. The stalk of the plant gets wider and wider until it gets big enough to support its own weight. Even over time, becoming a woody plant, they always start herbaceous and eventually becoming woody. But when we consider starches, we also notice that a lot of the things that we get food from with plants, think of the plants that you've eaten. Things like broccoli and even Brussels sprouts and lettuce and carrots. Each one of those pieces of that plant has a root, has a leaf, and has some kind of nutrients that are going into it. And every plant stores their starches differently. If you think of Brussels sprouts, we literally take the unbudded parts of the plant and we cut those off and we eat what the starches that are stored inside of the leaves of that plant. Lettuce the same. We think about carrots, we actually eat the roots of a carrot. Carrots store all that starch. It's a biennial, so it stores all that starch for its second growing season so it can grow and reproduce, and we eat the food from that. Our body uses those starches. But second, we actually use also glycogen. That's how we store these. So when we eat a food or sugar, our body will actually make complex chains of molecules. Don't remember if I have a picture. I do. We have a set of chains. Again, this one's nice and simple. They're just little hexagons. But this represents down here a whole chemical model, glucose chains. And so here's a glucose chain, glucose chain, glucose chain, glucose chain, glucose chain. Well, the whole thing, the storage molecule, is called a starch. Our body, when it's hungry, breaks one of those. We use one. We use another. We use another. And it takes time for us to use this. So we store it as starch in plants, and as people we store it as glycogen. Please be aware of that, mark it, note it, know it. The best source of carbs is plant-based food. It mentions in your book the best source of carbs, your diet. This is the right side of 182. Are starches found in plant-based foods, including grains, fruits, and vegetables. You got to think about it. If it comes from plants, it makes sense to eat plants to get it. Nutritionists recommend that most of carbs in your diet consist of polysaccharides, such as starch, rather than monosaccharides or disaccharides. The last one here we mentioned is another type of sugar called cellulose. Notice it is a polysaccharide, but it's so complex that it takes a long time and even is incapable before it has passed through our digestive tract of being absorbed for all of its nutrients. What is it? It's a polysaccharide that builds the walls of plant cells and gives them their structure. The polysaccharides that we cannot digest, specifically cellulose, are often called fibers. Fibers are known for their ability to help keep our colon and intestines healthy. Most of the time it's because we do not digest it. It is used as roughage to keep things going through the intestines well. And so you'll see that on the top left of 183. mentions that even one of these molecules can have several links. Unlike sugars and starches, cellulose is not digestible by humans and passes unchanged through the digestive tract. We mentioned because cows have the complex stomach systems that they do, that cows are actually able to digest more food from cellulose than even we can. All right, changing the topic, we have the first type was carbohydrates. The next things you're going to see is fats, but I have here the word on the screen, lipids. Well, this starts on page 183, and it mentions lipids are example here. It's a group of biological compounds characterized by insolubility in water. These are biological compounds, and they cannot dissolve in water. That's what the term insoluble means. If you were to take oils and you put it in water, they would not mix. They would not dissolve perfectly. These are fats and oils. And so I mentioned here, if you wanted to remember the term lipids and associate it with a fat, you would want to know that, if you got hit in the lip, you get that fat lip. And so fat lips, fats, lipids, or fats. Okay. Chemicals the body uses to store energy. Now, this is true. As you eat food, if you eat too much food for the energy need of your body, in an amazing way that God designed it to have, stores that sugar as a fat. If you, for some reason, weren't able to eat, your body would start to go to those fat and glycogen stores, pull from those for energy as needed, and it would be able to draw from those. There's other things that your lipids do, but that would be the main thing. Well, I want you to know that lipids are composed of carbon and hydrogen rods that form a thing called a fatty acid. So lipids are composed of fatty acids. And we'll talk through this in just a moment. One of the examples is this one called a stearic acid. All right, so here's an example of stearic acid. Basically, you have a hydrocarbon. It's why we in the last section talked about hydrocarbons and their complex shapes. I can't call this methane. It's got a long group, but you'll notice the end of this is a carboxyl head. And if something has a carboxyl head, it's called an acid. So in this case, we have stearic acid. And so that acid is the fatty acid that makes up an entire fat molecule. Page 183, you will see when you take 3 of those stearic acids, you can actually make a full molecule. Looks like a camera tripod there in your book. Mentioned fats, and this portion is important as well. A fat molecule consists of 3 fatty acid rods. Here we go. So we have a fat molecule. You can notice he's big, and so don't judge him, but he's a little fat. No pun intended. Actually, all kinds of pun intended. And so fat molecule here, you've got a bunch of chains. They look really complicated. If you have no idea as far as chemistry goes, this alone could be a really scary picture. This top portion is what we call glycerol. And then the bottom, this is stearic acid, stearic acid, stearic acid. Here's your carboxyl heads. And those attach to the glycerol molecule, almost like a hanger in your closet. And they kind of hang on this. This whole thing, when it is put together, this tripod is a fat, okay, chemically. You notice that they are efficient storage devices. So we go back here, and we said it consists of 3 fatty acid rods with their handles attached to a molecule of glycerol. There's the molecule, the chemical structure of glycerol. And I wanted to take some time and talk to you about saturated and unsaturated fats. Fats can either be classified as either saturated or even unsaturated. Saturated fats come from specifically animals. They're usually solid at room temperature. I don't know about you, but when you cook roast beef or a roast or something, it's an amazing thing. You put it in the crock pot. You've got the roast beef. You got the carrots. You got whatever else you got in there. But you got to have the beef and potatoes, carrots, and all that. It's great. But when you put it in the fridge, you'll notice the next day you pull that out, or even if it sits in the crock pot a little bit longer that day and it hasn't been heating, as it goes to room temperature, the fats from that beef, the cow that once was, will turn to solid. They'll actually turn white. There will be a chemical change there. We call that lard, and it mentions that is an animal fat. Usually this is solid at room temperature. Now, we will talk more about what this means here. Each carbon has a maximum amount of hydrogens attached, but it is saturated. Because the hydrogens have maximized, that's what we mean by saturation. It has all of its spots filled. I'll explain the chemistry in a minute. I want to talk about the biology for a moment. Unsaturated fats are kind of the opposite. They don't have all their carbons filled. That's the chemistry side. But they come from vegetables. Think about peanut oil or canola oil or one of those. And so liquid at room temperature have fewer hydrogens as a result of double bond between atoms. So why are they unsaturated versus saturated? It has to do with their chemistry. But let's start with this. Let's make a little comparison contrasting. Okay, saturated fats are those that are animal fats. They're usually solid at room temperature, whereas vegetable oil, you probably have bottles of this at home, they're not solid in your cabinets. They're clearly liquid at room temperature. And so you want to be able to contrast those. They're both fats, and they're both types of fats. One exists as liquid at room temperature, one exists as solid. So let's talk about the chemistry aspect of it. By the way, here's my fat molecule. He is quite large. I wanted to talk to you about the chemistry. Now this is a very pixelated picture, but let's see if I can talk through what's happening. Looking at a drawing, let's take a fat for a moment, and I think we can actually do this color. So we have a carbon, 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 carbon. Okay, and each one of these carbons is going to go off to an H. I'm not going to draw the H's for sake of time, but they're going to be full. Okay, and then the ones on the end have 3 H's that they go to. I'll draw these ones just for visual sake. The other ones have 2. So here's an H, here's an H, here's an H. Now all these slots also have H's. All right. So this right here, as we have it, now at the very end of one we'll have that carboxyl head, and so actually I'll remove this H for a moment. Because we'll have, for a fat, it'll be a little more complicated than this. We'll have a carboxyl head, so we have the double bonded oxygen, single bonded OH over here. All right. So we know that all of these slots represent H's, and they would go on and so forth. This right here represents a fat. Chemically, we know that this thing is going to be solid at room temperature. Okay, and from the chemistry statement, we said that this thing was saturated. Now, you know the term saturated just from being alive and experiencing it yourself. Something spills in the kitchen, you take a towel to pick it up, and as you put the towel down on your spill, you notice the towel starts to soak in the liquids. But if it's a large spill, as you put the paper towel down, it'll start to, as you pick it up, drip. And that dripping means it's saturated and cannot hold any more. So saturated means it cannot hold more. Okay, so what can't it hold? Well, it can't hold any more hydrogens in this case from a chemistry perspective. So now if we have all these hydrogens filled, okay, that's what we have. This is a saturated fat because it has the carboxylic head. It has all the H's that it could possibly have in every direction. So then the question is, what does an unsaturated fat look like? So again, here's the contrast. We'll just kind of draw a line here. Down here we've got C, C, C. Let's go to our carboxylic head. O, OH. We have the 3 H's over here. Okay, line, 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 line. Okay, remember, this is part of what's called a Lewis structure. Lewis structures have these dots, or Lewis dots, or even bonds that are drawn. I'll draw an H here. And they're supposed to represent a covalent bond, a share between carbon and hydrogen. So as I draw this, please understand that every single atom needs to have its octet full. So if I consider just this carbon, around it I'll notice that this line here represents 2 electrons, this one represents 2 electrons, and so as I go around we have 2, 4, 6, 8, so this carbon has 8 valence electrons and it is satisfied with an octet. Okay? It is possible though for me to decide that right here, different marker color, right here I could put a double bond. But the moment I do that, I change the number of bonds around this. Now if I start counting around the carbon that I'm looking at specifically, you will notice that now I have 2, 4, 6, 8, 10 electrons around this carbon. That's not an octet, that'd be like a dectet, and that's not a thing to my knowledge. Dec meaning 10. So what this means is, as I look around this one carbon in specific, I have 2, 4, 6, 8, 10. I need to remove one of these bonding hydrogens in order for this to have 2, 4, 6, 8. Well, now I have a slot where hydrogen used to be. Well, I only considered this carbon. On this carbon over here, the same thing applies. Notice we have 1, or 2, 4, 6, 8, 10. So I'm going to remove one of those hydrogens. So down here I have a slot where hydrogen should be. And so what we have here are holes that should have H's in them. So this one is unsaturated. So from a chemistry perspective, this thing is not full. It can hold more. So as we consider the H's that would be drawn on this molecule, we have all the satisfaction of knowing that that thing is pretty much full. But what determines whether or not this thing can be full or not? It's the presence of these double bonds. The second I form double bonds, I start taking away slots where H's can be. So the presence of H's, or double bonds, let me just say this, unsaturated has double bonds, and it could hold more, hence being unsaturated. So chemically, that's what an oil is, whereas a fat chemically is saturated and has all of its H's filled. But I hope you are familiar with peanut oil. Peanut oil and peanut butter aren't that different from each other. That was one of the amazing things about it. If you take your books to the next page, 184, I wanted to show you more of this. By the way, some of the stuff that I still have up on the screen, you can see some pictures in your book 184. If you turn there for a moment. On 184, you'll look at the very top, you'll see the saturated fatty acid, specifically stearic acid, excuse me. Same drawing that I have pretty much. But you'll notice the one at the bottom is a saturated fat. They use the term unsaturated, I'm sorry. The middle one there, monounsaturated fatty acid, you'll notice there's a double bond. So you'll notice the arrows point to one missing hydrogen pair. So it has a slot where hydrogen could be. It's unsaturated. Notice it says monounsaturated because it only has one pair missing. At the bottom it has a monounsaturated pair. And so we have this unsaturation that's occurring. Now there is another term that I wanted you to be familiar with, and I think that would be on the next slide, and it is the hydrogenation. This is the process of adding hydrogen atoms to the double bonds. Now, chemically, that's what's happening. Obviously, this process is more complex in a lab, but what we're doing is we're taking something like peanut oil and we're hydrogenating it, putting hydrogen atoms into it to make it double bonds fill. Actually, we're making single bonds now to produce an oil. Okay? To change that oil into a butter or a paste that could be spread. So it allows the oil to act rather than as something that would soak in, more as a spread. And so butter substituted could be spread rather than poured. So if you look at the ingredients of something like peanut butter, you'll notice this is made from peanuts and other things. Right here at the arrow we have, let's see where it is, hydrogenated vegetable oil. Okay, so there it is. So notice peanut butter is taking peanut oil, hydrogenating it chemically to form this. Okay. The next biological compound I wanted to talk to you about was cholesterol. You've probably heard of cholesterol as maybe something that is not necessarily a good thing, but all of these things are nutrients. It doesn't have to be a bad thing. This is page 184. You'll notice cholesterol is another lipid. It's a fat. It's used by the body to make bile. Bile is one of your digestive enzymes, digestive juices produced by our liver, stored in our gallbladder. Vitamin D, cell membranes, and even hormones. Hormones, again, controlling almost everything that your body does, and so cholesterol is important. It has a little molecule that carries it. You'll notice here lipo. Remember the prefix lipo referred to fats, fat lip, lipoproteins. And so they have this structure, this molecule that carries cholesterols called a lipoprotein. You need to associate that. Here's the definition. It's a globule. That's a fancy science term for a blob. Large cluster of things, of cholesterol encased in a membrane made of lipids and proteins. Lipoprotein is a lipid protein. Now, there is a problem with this. There are two types of lipoproteins. I will write these abbreviations at the top here. the terms are LDL and HDL. I want to talk to you about what this stands for. LDL stands for low density. So I mentioned they were globs or blobs. And this next one is high density. Okay? Okay? So low density is not as tight of a blob as high density is. And so it mentions in your book, although cholesterol is an important nutrient, a prolonged excess of one type of LDL, low density type, is in the bloodstream can cause heart disease and heart damage and artery damage. However, another type can actually reduce that. So HDL would be better for you. So we're going to go ahead and make an association. There are two types of lipoproteins. The one that's good for me is this one. This one right here, no bueno. Try to avoid that type of cholesterol that is in your body. The heart disease is a thing, and you want to take care of your body while you have it. High density lipoproteins can in fact reduce the risks of heart disease. All right, and the last thing I wanted to talk to you about is, oh, by the way, a couple pictures here before I move on. Chemically, here's the molecule of cholesterol. Again, all these rings represent stuff, but as a constituent of membranes and source of steroid hormones, or just hormones in general, for that. They also make every wall of every cell is actually made up of a phospholipid, which involves cholesterol going through them. So we do, in fact, need these. Here are the high-density lipoproteins. So lipoproteins are simply those containers, and you can see this LDL versus HDL, high density. There's a very small one. These are the only two we talked about. There apparently are other types. But here's LDL and HDL. HDL is much more dense, and that's what I wanted you to see. Proteins, and this is the last one for this section here. Proteins is the complex organic molecule used to build and maintain living cells. Knowing individuals who spend a lot of time in athletics, one of the most important things that they need to eat is obviously energy-based foods, the carbs and carbohydrates. It's something you want to eat to restore the energy after a workout, probably even before. But also, you want to eat proteins because they are used to build and maintain cells. A muscle is a lot like a rubber band, and over time that rubber band gets stretched, it gets worn, and as you use a muscle it forms micro tears, and those micro tears are part of how the muscle gets stronger and stronger, and you're going to need those. Okay, these are complex organic molecules used to build and maintain living cells, and so if your muscles start to tear, these proteins will come in and repair those muscles. Now the cool thing about muscles is when they tear and are repaired, they get stronger. So proteins indirectly make big muscles. You got to actually use the muscles to make big muscles. Remember the biology term for that is hypertrophy, the growth of muscles. So about 50% of the body's dry weight is in fact proteins. Proteins aren't just muscle making molecules. They're also used throughout your body to make the cell do what it needs to do and various other tasks. Proteins are also composed of C, H, and O, so similar to carbohydrates, but they also have nitrogen in them. They're a component in cell structure and even control some chemicals. One thing I want you to know about proteins, just like fats were made up of fatty acids, proteins are made up of amino acids. And so amino acids are the building blocks of proteins. When I say building blocks, you think of those little ABC blocks that kids play with. We're really thinking more like the rings of chemistry that are making up all of the long chains of molecules. There are about 20. So as complicated as chemistry is, there's only 20 different amino acids that can make all the variety of proteins that exist. Well, proteins are pretty cool looking. They're very complex. Remember the chains I showed you, like here's the linear pattern on the left, but on the right they would make something like this, and that would just be fun to draw on the board, right? But this is kind of what it would look like. A protein is just a blob or a globular cluster of atoms and molecules together. On the left here I have all of the 20 amino acids that exist, and it is these amino acids that would make the protein here on the right, and it would be a mixture of different ones. And so most of the proteins that you need actually can be gotten from the food that you eat. Mentions this page 186. Although the body can synthesize some amino acids, it can make its own. Some must be obtained directly from food because the body cannot synthesize them or create them itself. Such amino acids are called essentials. These are amino acids you have to get from elsewhere. Certain foods contain more essential amino acids than others. If you want protein, one of the easiest things to get protein from is something that once had protein. Mussels. animal muscles we call meat. And so it says here in your book, most animal sources of protein provide all the essential amino acids in adequate amounts. Thus, eggs, poultry, fish, red meat, milk, cheese, etc. are good sources of that.